All right, so guys, today we're going to talk about Metagenomics 101. And I just want to say, and I think all the other instructors for all the other days probably feel the same way. What we're going to do, your crash course today could legitimately be a class at a university. In fact, it is a class that we teach here. And so we really tried to take the highlights, but it also leaves some nuggets for the advanced kind of expert user as well. And so our real goal is that, and what we're so excited about with this course is that you're now going to be in teams and we're speaking across different audiences. And so we really tried to distill it down to the fundamentals of metagenomics. And that's what you're going to get in the next 40 or 35 minutes of your time with me. So just to orient yourself, myself and Alicia are going to do some crash courses for you. And then hopefully the knowledge that you get in this first morning session will actually be what you apply for those that are following online. And we'll have a similar format for the afternoon. And then after we've worked today, I really want to call everyone back because we have some great speakers, both from JGI, who are going to kind of talk about the resources and how, especially for you early career scientists, I think this will be extremely beneficial. And then we actually have Owen and Ulas from Lawrence Berkeley National Lab, and they're talking kind of a more advanced, like where do we take the knowledge that you've discerned and you've developed today, and how do you apply that in a more quantitative framework? And so I really wanted to kind of stress that the whole checking back in later on in the day after some of the breaks. I also wanted to give a shout out to the many people that really helped build today. From my team on the CSU side, graduate students and postdocs, you'll be hearing from many of them today. And then also on the K-base side, who are shown here in orange. And then Bridget and Sean did a lot of the legwork. Many of the narratives were actually built by Sean. They're not here today, but I just wanted them to be recognized. Okay, overall goals for today. We have three main goals. We're going to start with just basically this river microbiome adventure. And I really want us all to think about this as an adventure today because we're going to be approaching science today in teams, and we're going to be approaching it from all different backgrounds. And so the more open we keep our perspectives and the more open we are to communicating, the better off we'll be. As happens in any science team where you have cross disciplines coming together, multiple disciplines coming together, we're going to have a lot of basic fundamental vocab today, and there's just no way around it, folks. I know none of us love those things, but I really want to ensure that we're all speaking the same common language when we start discussing science. So, miracle microbes that fix the planet and our health. I mean, I think you'd have to live in a vacuum today to not hear this microbiome language. I mean, it's all over the news, talking about how microbes colonize our cells, the insides of our guts, and the world around us. And, they play essential roles in wastewater, in hydrocarbon remediation, in breaking down plastics, in increasing energy yield for crops, and even managing our human health. The fact of the matter, folks, is there is no miracle microbes. This is not new. What has changed is our ability to sample the microbial, this invisible world that lives on and amongst us. And the tools that you're going to be using today are essentially those tools, those advances. When we talk about the microbiome isn't just the microbes. Often we just think of it through a biotic lens, but I really want us to think the microbiome actually encapsulates the ecosystem. And so we're talking about the metabolites, the chemistry, and hopefully what you'll see today is the microbial perspective, but be able to link to the other abiotic and chemical portions of this world. It doesn't only focus on bacteria and archaea as well. There's fungi and protozoan, as well as viruses. So when we use this term microbiome, it's much more than just the microbes, which we often assume. The tool you're going to be using today is metagenomes, and this is really what's revolutionized the field of microbiome science. This is why we can listen to commercials about our guts and probiotics. This is why my dog takes probiotics. It's because we have a whole new understanding of the metabolic capabilities that are encoded by microorganisms because of this tool. And so we really wanted to expose you to common workflows that expert users use, but that anyone, hopefully in this framework, can also use. I saw that Alicia posted that the narratives will be available after today too. So those of you who are following along more remotely, I encourage you to check those out as well. Okay, so as Amy mentioned, we have sites across the globe that we're using as our model samples. You will either be in a group today that's surface water, meaning it's planktonic, and so it's the microbial community there, or you'll be looking at pore water that's basically extracted from the sediments. Essentially, when those samples are collected, they're filtered, and on those filters, we retain the microbial biomass. From those microbial biomass filters, we extract the DNA. And so this is what's changed, because historically, when we used to do microbiology, we would take the filters and we would go back to the lab and have to culture all the different organisms to know what they do. Now, in this idealized example here, you could imagine that we can just take the genomes out. And now we have this metabolic blueprint of each organism that's in that sample, in a perfect world, and all the things that organism does. And here's actually Leandra, one of our graduate students, sampling for the wonders, and you can see it going through the filter. And so the effluent that comes out of the filter is what we use for the chemistry. So the sites that we're going to be doing metagenomics on today are here in orange, and I'll talk about them in a second. But what I wanted to let you know is that you're part of a larger community, and I think Amy and James did a great job of kicking this off. And so what we're trying to do, and this is in collaboration with the Joint Genome Institute, which has been supported by a CSP, is builds this genome-resolved open watershed database. And so our goal is to sample the world's rivers and actually compile and do exactly the steps that you're going to be doing today, but do them across all of these rivers. And so you can imagine that we could have this encyclopedia of microorganisms that live in planktonic or water sediments and all the reactions they can catalyze. And we believe that this kind of ecosystem approach, but community-focused approach, is what we really need to start transforming metagenomics into the future and really enable the modeling and all the other things that you're going to be hearing about this week. And so this is kind of our proposed sequencing plan in collaboration with JGI, as well as with KBase, as well as with the NMDC, and completely enabled by Wonders. You can see that Wonders is a large portion of the samples. But we have many other PIs who are also donating and work in river systems and offering to give samples as well. And so if you're interested in that too, you can reach out to myself or James, who are kind of spearheading this effort. Okay, you kind of heard more about where we are today. You'll be either working in one of these 4 groups for the metagenomics today, and within those groups, you'll either be in a pore water or a surface water group. So that's to orient you. We have many people from many different backgrounds, and so I didn't want to take it for granted that we all instinctually knew what a gene and a genome was. And so the next kind of part of this is just to kind of give you some basic concepts and some framework for what we're going to be looking at today. So basically, a genome is the set of genes in a cell. And so, in this picture here, we have, 5 or so different colored genes, and they're organized. And collectively, these are the metabolic kind of powerhouse, or this basically is the metabolic blueprint of what that cell can do. And so if we think of microorganisms as these kind of catalytic sacs that exist in the environment that are doing all these transformations that we so care about in terms of carbon and nitrogen and sulfur, you can imagine if we could rebuild these genomes and inventory and annotate these genes in terms of their functional capabilities, we would have a really solid understanding of what microbes can do. And really, the reason why is these genes are basically encoded into proteins. And proteins are those enzymes that we really care about. And then when we think about the metabolites, we often think about the metabolites as what the microorganisms are either using as substrates and products or interacting with. And so you'll be looking at the genomes in this course today and metabolomes and kind of overlaying them as the week proceeds. So an analogy here, and then when we think about genomes, we think about it as those kind of old school recipe cards that your grandma has. And we think about the genes as the ingredients. And so you can see here that each one of these genes is used to kind of make up the recipe. However, what you're going to be hearing about this afternoon is that we don't really quite have a perfect understanding of these recipe cards. In fact, when we look at annotation, which is basically assigning a functional attribute to a gene, we find that instead of having brown sugar and white sugar, we have non-specific annotation. So really we can say you're going to add 3/4 of a cup of some sugar and 3/4 cup of another sugar. In a similar analogy, we also have many genes in a genome that are hypothetical. And what that means is that we can see a start for that gene and we can see an end for that gene. And we can even look at it in our databases and we can find other organisms that have matches to that. So it's conserved across life. Yet we don't really have any biochemical understanding of that gene. And that's a large portion of actually the microbial genome space. So instead of saying we're going to add 3 cups of flour, in this analogy, that would be like we're going to add a white solid powder. That could be powdered sugar, that could be white sugar, that could be flour, that could be a range of other things. And the last portion of the microbial sphere, which I think is also this very exciting space, is that there are many genes in a genome that are completely unannotated. That means, hey, it looks like a gene, but when we hit our databases, we don't know what it even hits to. We don't even know if it necessarily hits to anything else that we've seen. And so instead of having 3 eggs, we would basically be like, we don't know if this is a solid or a liquid. And so this example actually exists in workhorse E. coli. I think we know that organism. It's a model workhorse for microbiologists. And even in E. coli, there's about 30% of its genome that would fall into this hypothetical or unannotated, and maybe even non-specific annotations. So keep that in mind. I just wanted you to have in your mind the caveats. It still means that maybe 50 to 70% of the genome we can see and we can assign functions to. So all hope is not lost. But there is this space that we're still, it's always one of the challenges we're up against. You're going to learn about metagenomics today, which is basically a collection of genomes from the environment. So in an idealized world, you would have, for every cell that you would have a near complete genome for. At the end of today, we'll have probably a more realistic understanding of this. But you could imagine then you would have many, many genomes. But if you couldn't see the flour and you couldn't see the salt sugar and you couldn't see the eggs, understanding what those organisms do becomes a lot more challenging. And that is something you're going to see in the space this afternoon when you're working with your genomes. Okay, so the morning agenda, this is where we are. We're really trying to prepare you for how you go from raw reads, which is the raw data, to genomes. And this is ambitious, like I said. So we're going to do this in 3 steps. We're basically breaking this down into 3 steps. We're going to be talking about how we work with the reads, and then we're going to talk about how we put these reads into larger fragments. And then we're going to actually talk about how we take those larger fragments and cluster them into genome-like organisms. And so that's the overview for the last half of this talk. Okay, surface water, pore water, like we discussed. At the average genome, just to give you a size, because we're going to be talking about base pairs, which is basically the single nucleotide in a genome length, in a nucleotide sequence. And a genome, I just wanted you to have a context, something like E. coli is maybe around 3 million base pairs. When we take that biomass that we take off the filters, we actually fragment that biomass so that we can put it on the sequencer. We fragment that down to about 500 base pairs. When we get the data out of the sequencer, it's called short reads. And the reason why it's called short reads is those are about 150 base pairs. And so you'll actually be looking at the quality of 150 base pair, and you'll be looking at each base pair position. So you'll have a kind of a more intuitive understanding for what this data actually looks like after doing the narrative today. So we fragment the DNA. An artifact of doing sequencing is that the sequencing technology itself adds on adapters and indices. And so we don't just take the raw, even if our quality looks good, we often don't just take the raw data. We typically work it through a program to trim off kind of those artifacts that were added during the sequencing process. And when you receive sequences, you're going to receive basically a forward sequence. It's going to have a gap or an insert size, and then a reverse sequence. And so a lot of the analyses you're going to be doing today are going to be working with forward and reverse reads. And I just wanted to kind of orient you to this. If you're interested into sequencing, I know I kind of went over it pretty quickly. There's a nice YouTube video at the bottom that kind of goes through the kind of sequencing, the Illumina sequencing that we're using today. And that's the NovaSeq. Okay, so when we get our reads back, basically, we wanted to take you through the exercise like you were just getting the data, just like we do from our sequencing facility. The first thing we do is we check the sequence quality. Actually, the first thing we do is we check the sequencing amount. Because you pay for a certain amount of sequencing. And so the first thing you want to know is, I paid for 12 gigabase pairs of sequencing. Did I receive that amount of sequencing? And so the tool that you're going to be using, or the application that you're going to be using today to do so, is FastQC. Oops. So in FastQC, you're going to basically look at the summary of the quality and the amount of reads that you have off the sequencer. And so FastQC has a forward. Basically, you have a report for your forward and your reverse reads here. And so this is, I'm just showing you one of the reports, but it tells you the total amount of sequences you have, and it actually gives you the sequence length. This is some of my really old data. The reads that you're going to be working with today are about 150 base pairs. So the first thing we ask you to do is exactly what I ask my students to do, which is, did we get the sequencing that we paid for? Can you confirm how much sequencing we got? And so to get that, you're basically going to take the total number of reads, which is going to be double this amount, because this is just the one of the forward and the reverse. And then you're going to multiply it by the read length. And then you're going to divide it by 10 to the 9, and that'll give you the gigabase pairs of sequencing. And so we want you to look at how much sequencing you have per sample, because our ability to understand which microbes are there and the jobs they do are often dictated about how extensively we sample the community. And so the more sequencing we have, the better apt we are at actually recovering those organisms and those processes we care about. And you should find that your sequencing is probably in the range of 20 to 40 gigs today. The next part is read quality. Again, this is older sequencing, and I chose this on purpose because our read quality is actually improved quite a bit today. But what you can see now is this is actually this is every base pair position of your read, and then on the x-axis, and the y-axis is the quality. So obviously we would want our quality to be in this green region, but you can see a large portion of our reads actually fall into these there's a lot of junk on those reads. And so what you're going to do is actually trim those reads, and the tool that you're going to use to do that is in KBase. It's the app is Trim-O-Matic. And then what we're going to have you do is actually compare the reads prior to sequencing and after sequencing. We want you to note how many of your reads did you lose, and what now does the overall quality of your reads look like. You start applying and using those reads later to look at how abundant certain organisms or certain processes are, is having nice quality reads to work with. I will say that if you collaborate with JGI, which you'll learn more about in this evening session, they actually do this oftentimes for you, and you can download your trimmed, cleaned up reads. However, many sequencing facilities do not. And so if you're in that case, we wanted to expose you to these opportunities. Okay. Assembly. This is the art of what we're gonna be doing today. And so in assembly now, we have all these 150 base pair reads. You just saw the quality of them. And now we're going to take those and we're going to basically start stitching those together. And what we're stitching them together to get is contiguous sequence. So we want what we call contigs. And that stands for contiguous sequence. And so really we want to achieve contigs greater than 1,000 base pairs, but really more on the order of 500,000 base pairs or 100,000 base pairs. It is possible to take trim reads from a microbial community, assemble them, and obtain closed genomes. There's many examples in literature today where we can do this. The reason why we hear so much about the microbiome today, in my opinion, is partially due to the advances in the assembly software and technology. So these computational advances has really made it possible for us now to go into the environment and pull out those near complete genomes of organisms that are doing a lot of the heavy lifting in terms of the biogeochemical cycling that we so care about. So really the goal of assembly is to take these small little pieces and represent an entire genome and assemble and build them into this large masterpiece. However, when we assemble, we do what's called de novo assembly, meaning we don't really have a guide. I mean, loosely we have a guide because we sort of know the general features of a microbial, especially bacterial genome. We have a general sense of the size, but we don't really know how all these pieces come together. And so it's very similar to doing a puzzle with a blurred out kind of box cover. So you kind of know the gist, there's some elephants in there, but you don't really know how everything may come together. And that's really what we're doing when we're doing this assembly. Oh, okay. Just like doing a puzzle, there's actually many analogies to some of the concerns or things to keep in mind when we do an assembly. For example, in this puzzle that's partially done here, I'm showing you a starfish. And what you'll find also, and I know many, is that you're locally accurate. You'll find that you'll work on this little piece and you can really assemble it well, and like the case of the starfish. But you don't have larger context for where that piece goes in the whole overall system. And that happens many, many times when we do assemblies. So we say they're locally accurate. We might get a contiguous sequence or a fraction of contigs, which really come together nicely, but we can't fit them to other parts of the genome. The hardest part to assemble are repetitive regions. And so you guys know from doing puzzles that the worst thing you can get is like a solid blue sky, or in this case, this bright yellow, solid one color, no words, no text image. And the reason is those repetitive regions get a lot, you lose that context for how they come together. And so you're forced to only rely on a limited or less amount of information. And we find that's true in genomes. So when we want to build up metagenomes or summarize the whole encyclopedia of genomes in a community, we're often hamstrung by the parts of the genomes that are really repetitive. And so just like puzzles, genomes can be very complicated. Just like genomes, however, we can use this idea of insert size between our reads to kind of contextualize us and figure out how things come together. So the example I give here is this fish in the bottom. Once you start working on that fish, maybe you're on the tail of that fish, you have a sense, an intuitive sense from your a priori knowledge of how big that fish is going to be. And similarly, these contigs come together in a similar fashion. So we can use kind of knowledge prior, either insert size or contextual or Sydney or other things, to help us put these fragments together. Unlike this puzzle, which would be completely unsatisfying if this was our final product, in metagenomics, if this is an ecosystem metagenome, I would say maybe it's about 70% complete. That's a metagenomics. You can get a gist for what the message is. You can see that it's a tropical theme. You have a sense for how big the puzzle is. You have a sense for some of the metabolic capabilities. There's a starfish, there's a fish at the bottom, there's a volcano. So we can do a lot with little. So I don't want to present it as all doom and gloom today, but many of the genomes that you're going to be working with today are going to be unfinished. And that is one of the things you're going to learn how to do, is basically, how do I assess the quality of my genome? Okay, so there's two broad ways to do metagenomics. Metagenomics means different things to different people, so I want to make sure I define it clearly. We are not going to be using a read mapping approach today. That means we take the reads and we just map them to a database. It is computationally very fast. It gives us a sense of what's in our data set that looks like what's in the database. But we miss a lot of novelty when we do that. Instead, we're going to be using an assembly-based approach, which I just described. Essentially, we're going to stitch together all those little reads and make different length contiguous or contig sequences. Within the assembly-based approach that you're going to be doing today, throughout this week, you're going to be using two types of assemblies. You're going to be using the unbinned assemblies, which basically means we take those contigs that you see here. Actually, I'll use my thing. And you can imagine this blue contig. Let's say that's 5,000 base pairs, or about 5 genes long. And then we could annotate, basically we could call the genes on that contig and we could tell you, hey, that contig has the capacity to reduce nitrate. The next contig, it has the capacity to oxidize ammonium, so on and so forth. So in our bulk level metabolic potential, we take all of your contigs and we annotate it, and we have a good sense of the functional capabilities that are in your microbial community. In contrast to the unbinned approach, the binned approach is just one step beyond that. We now say, hey, look at this figure. We have a lot of these orange contigs. I bet these orange contigs all go together. They have very similar sequences, and in fact, there might even be some reads that connect them. And so we can reconstruct, or we can bin these into genomes and really get at more refined definitions. And so in the case of this green organism here, now it's possible to not only say, hey, this organism has the contig that is capable of denitrification, but it also has the capacity to use acetate as an electron donor. It can use hydrogen and CO2. It produces antibiotics. It's sensitive to these conditions. And so by binding to an organismal level, it gives us more coherence on what are the chemistries that could control denitrification in our system. If this green organism is really active in our system and the one driving a lot of that denitrification, understanding what are the constraints on that organism become really important. The drawback of the bind approach, which will become clear this week as you work with things, is that not all of the contigs go into the bins. And so that data reduction means that sometimes we miss key functionalities. And so I think that's why today you're going to be approached to the bind approach, but we'll parallel that with the unbind approach and the functionalities between the two. So you get a real sense for the advantages and the disadvantages of both methods. My goal is not to promote any one assembler or any one binner. In fact, I think one of the greatest attributes of KBase is that they don't either. And so there's many, many different apps for you to choose from. In today's narratives, we chose specific applications because we knew that they were optimized for these samples, but also two, in terms of energy or in terms of memory and other features for kind of doing a high throughput class. I will tell you, if you go through my lab's papers or other labs' papers, many times we use all of them. And so there is not necessarily, everyone always wants what's the one way, the one way to do metagenomics or the single protocol. And I'll tell you that this is the art aspect of this, is that you do it every single way and try to compile the most information you can. The tool we're going to be using today is MegaHit, but there's MetaSpades and IDVA-UD are also available on KBase. And we use them all equally. So in KBase today in your narrative, now you've gone from your reads, and we've cleaned up all your reads, so we know we're using good quality information. We're now using MegaHit to take those reads and make them into contigs. So we're going to lengthen those, basically stick a bunch of reads together and make these longer sequences. When you're using MegaHit, MegaHit has two settings, and I'm only telling you this because I want you to pay attention to the larger assembly. So MegaHit has meta-sensitive, which is really nice for smaller data sets, and it's a little bit more optimized, or it has meta-large. If we could, we ran both of your samples with each of those. So you might want to compare between the two. Do you get better assembly? Do you get better information using one over the other? However, for many of your samples, there was too much information and we actually used only the meta-large. For the questions today, we want you to use the meta-large assemblies. Also, as a note to people following along and also in the narrative, due to the fact that we have a lot of folks on KBase, we actually ask that folks don't run additional assemblies. So assemblies are memory, these assembly tools especially use a lot of memory to operate. In fact, this is probably the biggest handicap to folks doing metagenomic workflows. So oftentimes for the amount of sequencing that we're talking about in these samples, you would need access, if you weren't using KBase environment, to some high performance compute time. And typically the wall time is anywhere from 7 to 14 days for a single assembly. And so I just wanted to give you context for that so that you are aware of these tools and how intensive they are. We're going to use Quast to visualize how effective our assemblies are. The things I want you to look at are what's the distribution of my contigs? Contigs that are about 1,000 base pairs, that's about a single gene. It's not super useful for us. It's really hard to get information on who that organism is or what that gene links to in any sort of context. I really like to work with contigs when they're above 5 and ideally 10,000 base pairs. But right here in Quast, you'll see a distribution of your contigs across the different criteria. I mean, the largest contig here is a fragment of a genome. I mean, this could be a fourth of a genome if it was a small genome. So you could have assembled, basically 400,000 base pairs, which is a huge swath of that microbial genome. The other piece of data that people often look at when they're trying to assess how good is my assembly is this N50. And basically, this is just a weighted median. It basically lets you know where 50%, the length that 50% of your contigs fall above or below. It's another metric of distribution of contig length. Personally, I like to use kind of the histogram here. I like to know that I have a lot of contigs above 10K. but others report this. The last criterion you're going to want to think about when you're looking at your assemblies now, so you're looking at your contigs, is how much of my data went into that? And so typical numbers are anywhere around the order of 25 to 75%, depending on the community, depending on the data reduction, depending on a lot of different things. But it's something as a metric for if you're going to be fine-tuning your assemblies or using other assemblers, it gives you a sense that, hey, this assembler not only had larger contigs, not only had a greater proportion of larger contigs, but also recruited, 50% more of, that's an exaggeration, but 50% more of the raw data. And those are criteria just to give you a sense for how we start assessing performance of this tool. Okay, and I really think binning is my favorite part, but also the most elusive, probably intuitively, for some folks from teachings I've done previously. So now we're going to be talking about going from these contigs and putting these orange fragments all together to make a genome. So how do we take these little fragments and stick them together? Fortunately for today, because we're very limited on time, you don't actually have to learn the nuances of how these binning algorithms work. But along with assembly technology, binning technology has improved drastically. So if the first metagenome papers from the environment, it was actually from the Bamfield lab in Wrighton, Colorado, used this short read sequencing to reconstruct hundreds of thousands of genomes. And that paper was in 2012. Since that paper has come out, these technologies have advanced tremendously, making it so we can achieve bins in almost every ecosystem. What the bins give us, as we discussed earlier, is it allows us to link the red bug and its name to a certain function. It also allows us to look at the metabolic repertoire of that red bug and the other constraints on that metabolism that may be dictated by other nutrients or other resources that are beyond the capacity to oxidize methane, for example. Okay, how do I assess how good my bins are? So we're going to use completion and contamination today. This duck represents a genome from a pure cultured isolate. When you look at this duck, you're going to basically say, what are these duck? And you're going to inventory those. So you would say a complete duck genome is defined by having one beak, two legs with webbed feet, two wings, and a feathered tail. If you found in your bin a furry tail, a snout, a claw, a paw, you would probably conclude that there is some sort of contamination of a contig from another organism that's in your bin. And those would be really obvious contaminants. They're in fact the best contaminants to get because they're pretty easy to pull apart. So what this is showing you or attempting to articulate is that binning is not a perfect science. In fact, it's rather imperfect. And so there's a lot of quality control that happens at that binning level. But now if I told you your duck you actually have a metagenome and your duck is actually only 50% complete. Instead of having the full duck information, you only have 50% of the information. And now when you look at that duck bin, you see that or duck-like bin, you would see I have a beak. It's pretty compelling. It looks like a duck beak. How do I have four legs? Maybe I have some contamination. I don't know what's going on here. Oh, and because it's incomplete, I haven't sampled the wings and I don't have a tail. So this duck is actually not in fact a duck. And so this is the importance of kind of taking the time and using and creating mags that are as good of quality as possible. And so binning, again, there's many different tools. There's many different binners available on KBase that you'll have a chance to play with during the week. You're going to be using Metabat, and then you're going to be using CheckM. And CheckM is this tool here, which basically assesses the quality of a bin. And so you can see here, we're looking at this one bin, bin 007. You're going to have basically a report that you're going to access through this CheckM table. And then you're going to look and say, hey, this bin is 90% complete. That's pretty great. And it's 3% contamination. You can then use this comparison to duck criteria or known marker sets and say, what is the distribution of my known genes that are duck genes to this? And so you have 0, you're missing 27 of the known genes. You have 274 of the attributes. And for some of the known attributes, you actually have doubles. Okay, I've gone through this all really, really fast, I recognize. Our goal is just to introduce you to the vocabulary. You're going to be in teams today, and there's going to be a TA there. So I understand that you've had a 30-minute crash course, and a lot of vocabulary is going to be new. So I wanted to take a step back and just reorient ourselves to the noun and verb pairs that we're going to be using today. DNA is sequenced into reads. The reads, which are 150 base pairs, are assembled into contigs. The contigs can be anywhere more than 1,000 base pairs, typically. We like them even greater than that at like 10,000. Contigs, those fragments now of genomic information, can be binned into bins or metagenome assembled genomes, or MAGs. A noun is a data type. A verb is a process that converts a data type. In K-base parlance, nouns are going to be your data objects, and Alicia and your TAs will show you where those data objects are. In K-base parlance, the kind of tools or the verbs that we're going to be talking about, those are apps. And there's many different apps for a single verb. So we discussed there's many different assemblers and there's many different binners. Okay, so I wanted to fill you with the promise that metagenomics brings, especially for those of us working in environmental science. It provides us a way to catalog and inventory and look at the distribution of these functional traits that are so important and can be linked to hydrology as well as chemical data. But it also comes with caveats, and I hope I've done right by my field to articulate some of those. We get incomplete recovery, we often have pieces missing, and sometimes we are misinformed or have contaminating binning. And so the 3 workflows, and then I just wanted to give a shout out. This morning is kind of an overview of reads to bins. And the afternoon session is really the kind of ecosystem context that I think many of you will really enjoy. That's now how do we take those bins and see the functional capabilities within them? And how do we put those functionalities into an ecosystem context? And if there's some time, I think I'm happy to address questions or concerns.
Thanks, Kelly. So you got some thanks from the audience in the chat, and also some great questions. A lot of those questions have been being answered by your team in the background, which is awesome. One question that just came out that you could address is, what is the acceptable level of bins during assembly?
So the bins, obviously, it's going to be dependent on the structure or the composition of your microbial community. So today, the range is of anywhere from 12 to upwards of 25 or so bins from a single assembly. When we optimize data from these samples, we have about, I think, across the 2018 Wonders data, in terms of unique bins, we have about 350 unique bins to give you context. But again, I work in shale systems, and we have 3 bins from those samples, where when you work in the human gut and we just do a single assembly, we can get 150 bins. So it just depends on the composition of your microbial community.